Good day, Louis and Gems. Welcome to the first part of the discussion on genetics and heredity. This video is focused on Mendelian genetics or the works of Gregor Mendel. It includes his experiments on pea plants and the result of these experiments that led to the formulation of essential laws which served as anchors to the study of genetics and heredity. But first, let us unlock some terms that will be frequently used in this video and in this lesson. First, we have characteristic or character. These are heritable features that vary among organisms. For example, we have height, hair color, skin color, and blood type. Next is trait. Trait is a variant for a character. For example, tall, short, dark skin color, blonde hair, or even blood types, blood type O, A, and B. Next is gene. Gene is the basic unit of inheritance or the factor that controls expression of traits because it contains the information needed to specify a trait. Next is allele. Allele is a version of a gene and it's usually represented through letters. It comes in two or more forms, just like the blood group, but for us humans, as diploid organisms, we only have two alleles, one from the father and one from the mother. Let's take this one as an example. For the character blood type, we have these traits. We have blood type A, B, and O. Of course, we still have blood type AB, but it will be discussed on non-Mendelian genetics further. And the alleles for blood type, we have this one, and this one. We represent the alleles for blood type using capital letter I and a superscript of A for blood type A, and for blood type B, capital letter I, and a superscript of B, and then for blood type O, we have the small letter I. For blood type A, the alleles come in two sets. It can either be like this or like this, depending on what we have inherited from our mother or father. For example, for this set, it could be that we inherited both alleles of blood type A from the father and from the mother. But for this type of allele, for this set of allele, it could be that we inherited either blood type A allele from the father and O from the mother or vice versa. For the next terms, we will be using this example. Phenotype. Phenotype is the physical characteristic of an organism, while genotype is the genetic makeup of an organism. In this example, the phenotype is blood type A, but the genotype or the genetic makeup is this one. It has an allele for blood type A and allele for blood type O, but what is seen is only blood type A. Why? Because blood type A is dominant. Dominant traits are traits that are always expressed, and recessive traits are traits that are covered up. They are not necessarily deleted, they are just covered up and they may show up in the latter generations. So in this one, since blood type A is seen, blood type A is dominant and the allele for type O is recessive. We also have homozygous and heterozygous traits. When we say homozygous, these are the traits consisting of two similar alleles, while heterozygous traits consist of two different alleles. For this particular set of blood type A, the set of alleles are different. Therefore, this is an example of a heterozygous allele. Gregor Johann Mendel is the father of genetics, but before he was recognized as the father of genetics, he was first a botanist, a teacher, and a monk. During his time, people believed in the theory of blending inheritance for the reason that male and female parents are involved in producing offspring, so each of them contributes a factor to be inherited, and that these factors will be blended in their offspring. For example, a tall male and short female will have an offspring that will inherit a height that is intermediate to the parents. It may seem reasonable, but only when Mendel presented his work that they began to change their view about how traits are passed from parents to offspring. Although his works have not been easily accepted before, it has now given the fundamental laws of inheritance we all know of. One of the gifts Mendel received when he took charge of the monastery garden was a stock of peas, purebred peas developed by earlier gardeners. He later on decided to use the pea plants in his study. Garden pea or Pisum sativum has gained Mendel's attention due to certain factors. First, they are easy to grow or they have short life cycle since they are annual plants, meaning he will not have to wait for so long to get the results. Second, they are easy to pollinate. He can just cut the male parts of the flower and then dust the pollens from one plant to another to initiate cross-pollination. Next, it produces many offspring, therefore yielding more reliable results. And, of course, pea plants have observable traits with contrasting forms which served as basis for his data collection. These are the contrasting traits Mendel observed. We have the seed shape. For seed shape, round and wrinkled, seed color, yellow and green, flower color, purple and white, pod shape, inflated or smooth, and then constricted. For pod color, yellow and green. For flower position, axial and terminal. When we say axial, it means that the flowers are growing out of the leaf angle or near the stem, while terminal means that the flowers are at the ends of the stem and branches. For the stem height, we have tall and dwarf or short. Remember that in his first experiment, he had true breeds or pure breed pea plants, meaning if these plants were allowed to self-pollinate, they would produce offspring that were identical to themselves. In Mendel's first experiment, he crossed the contrasting traits of each character as his P generation or parent generation. We have yellow versus green, round versus wrinkled, green versus yellow, and so on. And he came out with these results under the F1 generation. As you can see, only one trait appeared or has been expressed. He then had two conclusions. First, each of the characters are controlled by a factor which we now all know as genes. Each of the characters he studied was controlled by one gene that occurred in two contrasting forms. These contrasting forms produced different traits. For example, a gene for seed color occurs in yellow form or green form. The different forms of a gene are called alleles. Alleles usually come in two forms like in pea plants, but some characters have more than two alleles, just like the blood type and skin color. For pea plants, the alleles for seed color, we only have yellow and green, while in blood type, we have the alleles for blood type A, blood type B, or blood type O. In this example, the alleles for yellow seed color are represented by two big letter Ys, one from the male parent and one from the female parent. We usually make use of the first letter of the dominant trait when we cross two parents or when we use these alleles for the Punnett square. So in here, the dominant trait is yellow, so we are making use of letter Y. An example of monohybrid cross for Mendel's first experiment will be shown on the next slides. The second conclusion on Mendel's first experiment is the principle of dominance, or principle of dominance and recessiveness. Some alleles are dominant while others are recessive. The dominant traits are always expressed and recessive traits are covered or seem to disappear on the phenotype of the offspring. When the gene pair is heterozygous, only one of the two genes is physically expressed. That which is expressed is dominant, while the other that is hidden or not expressed is recessive. This is not considered as a law because it's not true to all organisms. Other organisms exhibit codominance or incomplete dominance. All these will be discussed on the video about non-Mendelian genetics. With the result of his first experiment, he proved that blending inheritance is not true because there are dominant and recessive traits, and only one trait will be expressed and seen in the phenotype of the offspring rather than the intermediate of the parents. Since all of the F1 generation showed dominant traits, he again became curious and asked, what happened to the recessive traits? He then performed another cross and allowed the hybrid pea plants, or the F1 generation, to self-pollinate. The result of this cross produced the F2 generation, or the second filial generation. The phenotypic ratio of the offspring is 3:1, where there were 3 yellow seeds and 1 green seed, 3 round-shaped seed and 1 wrinkled seed, and so on. This is true to all of the characters he tested. He then became certain that recessive alleles did not disappear because they were showed on the F2 generation, and that they were just masked up by the dominant alleles. But how did the recessive alleles get separated with the dominant trait in the F2 generation? Mendel theorized that during the formation of gametes, the alleles were segregated or separated from each other. This can be proven by the use of a Punnett square. Going back to the first experiment, when Mendel crossed the contrasting traits of each character as his parent generation, yellow versus green, round versus wrinkled, and so on, and then he came out with the results under the F1 generation. We have yellow, round, green, inflated, axial, purple, and tall. For the parent generation, he crossed homozygous yellow and homozygous green seed color. This represents the segregation of alleles where they randomly combine with one of other parent's allele to predict the possible alleles of the offspring. All of the offspring for the first filial generation or F1 generation came out to be yellow. The phenotype of the F1 generation is 100% yellow seed color, and the genotype is 100% heterozygous yellow. In here, we can safely say that yellow is the dominant trait for seed color and green is the recessive trait. Same is true to the other characters. We can also make use of the Punnett square to predict the offspring of the crosses. Just draw a grid of perpendicular lines or have 9 boxes for the monohybrid cross or the cross involving only one character. In this example, we have the parents homozygous yellow seed and homozygous green seed. In here, we are only going to observe the seed color. That's why we are only going to make use of this Punnett square, a monohybrid cross Punnett square. So to get it started, individually, we place the alleles on each box. So we have the big letter Y here and another big Y here and small y's on the side. So this represents, assuming that this one is the set of alleles for the mother and this one is the set of alleles for the father. To plot the possible offspring, we write down on the middle box the combination of the alleles. So we have one big Y and one small y forming a heterozygous allele, and then you do the same for the rest. So big Y and small y will form heterozygous yellow. We always write down first the big letters or dominant alleles. So the genotypes are 4/4 heterozygous yellow, and the phenotype is 4/4 or 100% yellow seed. You can try plotting on your notebook the other characters and confirm the results of Mendel. You will get same results, 100% genotype and 100% phenotype for the dominant trait. Let us try to see the segregation of alleles in Mendel's second experiment. In here, I am also showing you the result of the first experiment where we have gotten the parents for our next cross. To see what happened to the recessive traits, Mendel allowed the hybrids to self-pollinate. An example is a hybrid plant having yellow seed colors. The result for this cross produced the second filial generation of offspring, and the ratio was 1 is to 2 is 1 for the genotype. One homozygous yellow, two heterozygous yellow, and as you can see, one homozygous green. So a recessive trait was expressed. The phenotypic ratio is 3:1, where there were 3 yellow seeds and 1 green seed. Here is another representation of the second experiment of Mendel using a monohybrid Punnett square. Alleles, these alleles are segregated individually and then combined or crossed with the other parent's alleles to predict the possible alleles of the offspring. As you can see, I have underlined this allele and this allele to show that these are small letters since they are hard to distinguish from one another. This cross shows the following genotypes: 25% or 1/4 homozygous yellow, 50% or 2 heterozygous yellow, and 25% homozygous green. And the phenotype is, of course, 75% yellow seed color, 50 plus 25%, and 25 green seed color. After the second experiment, Mendel marked the following conclusions. The presence of the dominant allele had masked the expression of the recessive allele, but the result of the F1 cross shows that some recessive alleles had managed to get away from the dominant alleles. F1 plants have one dominant factor and one recessive factor. The two factors segregate from each other when reproductive cells are made. Segregation of alleles occurs during gamete formation so that only one gene of a pair is received by each gamete or each reproductive cell. The alleles are paired up again when gametes fuse during the process of fertilization. That's why we only have two sets of alleles, one coming from the mother and one coming from the father. After settling his previous question and concluded the segregation of alleles during gamete formation, he wondered if the segregation happens independently. So he then asked, does the gene that controls one trait have anything to do with the gene that controls a different trait? Or does the gene for seed color have anything to do with the gene for seed shape? Mendel then theorized, if the genes are not connected, then they should segregate independently or undergo independent assortment. If the genes assort independently, some of the genes produced by the F2 plants will have new combinations of traits. He then performed his third experiment and tried to find answer to his curious question. On his third experiment, Mendel performed a cross to observe two traits, and here's a representation of the experiment. Since two traits are being considered, this is called a dihybrid cross or two-factor cross, and we will be needing a bigger Punnett square. We are going to use seed shape and seed color, and we will cross purebred round yellow seeds and purebred or purebred wrinkled green seeds. We are now seeing homozygous dominant alleles versus homozygous recessive alleles in the parent generation. Before plotting the alleles, they will be segregated such that each set of parent allele has an allele for seed shape, letter R, and allele for seed color. The segregated parent alleles will each be plotted on the Punnett square, and in writing the combination of the alleles of the offspring, the alleles for a character should be adjacent and the dominant allele should always be written first. For example, in our first box, the combination of a round and wrinkled seed will yield a heterozygous round allele, and the same is true with the color. Just like in the previous experiments that we had, the F1 generation produced 100% heterozygous round and yellow seeds, and the phenotype is 100% round and yellow. In here, all round seeds are yellow. The F1 offspring is not enough to answer Mendel's question, so another cross has to be done. To get the second filial generation, he allowed the hybrid to self-pollinate. The parent alleles would be heterozygous round yellow seeds. To get the F2 generation, these alleles will be used. The F1 alleles must be segregated before they get plotted to the Punnett square. In here, you can see remarkable difference of the segregated F1 alleles to the previous parent alleles. We are now ready to fill out the Punnett square. As you can see, the F1 alleles are no longer similar to one another. Therefore, the possible alleles of the offspring of the F2 generation varied significantly. We now have the following genotypes: 1/16 homozygous round and homozygous yellow, 2/16 homozygous round and heterozygous yellow, 2/16 heterozygous round and homozygous yellow, 4/16 heterozygous round and heterozygous yellow, 1 over 6 heterozygous round and homozygous green, 2 over 16 heterozygous round and homozygous green, 1 over 16 homozygous wrinkled and homozygous yellow, 2 over 16 homozygous wrinkled and heterozygous yellow, and 1 over 16 homozygous wrinkled and homozygous green. Going back to Mendel's question, does the gene that controls one trait have anything to do with the gene that controls a different trait? Or does the gene for seed color have anything to do with the gene for seed shape? In other words, should all round seeds be yellow? Let us look at the representation of the phenotype to see clearly the answer. This represents the phenotype of the F2 generation of our two-factor cross. There are 9 round yellow seeds, 3 green round yellow seeds. We also have 3 wrinkled yellow seeds and 1 wrinkled green seed. He proved his theory right and concluded that genes are not connected and they segregate independently or undergo independent assortment. One trait does not affect the other traits as seen in the experiment because not all yellow seeds are round and not all wrinkled seeds are green. Mendel's work can be summarized in 4 basic statements. First, the factors that control heredity are individual units known as genes. In organisms that reproduce sexually, genes are inherited from each parent. In cases in which an organism possesses 2 forms of the gene for a single character, some forms of the gene may be dominant and others may be recessive. The 2 forms of each gene are segregated during the formation of reproductive cells, and finally, the genes for the different traits may assort independently of one another, and that is also known as the law of independent assortment. You may also try answering the following problem sets to check your understanding about Mendelian genetics and, of course, the use of Punnett square. You may compare your answers with your classmates and friends, and you can also send your answers through the group chat or using the LMS. Thank you so much for watching.